Hello, everyone. Good morning, good afternoon, and good evening. Mark Estigribia here, Managing Director from MSQ Ventures based in New York City. We are honored to host Dr. Jin Li, Founder, Chairman, and CEO of HitGen, to our MSQ CEO Founder Webinar Series. Chengdu-based HitGen is a leading Chinese biotech company specializing in drug discovery, using its world-class DNA encoded library DEL platform. Founded in 2012, HitGen collaborates globally and advances innovative therapies across multiple disease areas. HitGen is listed on the Shanghai Stock Exchange's STAR market with a market cap of around $900 million, up 24% year over year and up 23% year to date. In 2024, HitGen reported revenues of approximately $60 million with healthy net profit margins of 15.4% and 22% for EBITDA. Dr. Jin Li, HitGen's founder, chairman, and CEO, is a recognized expert in DEL technology with international pharma experience. Under his leadership, HitGen has become a pioneer in early-stage drug discovery and global partnerships. Again, MSQ is based in New York City and our audience is global, spanning 20 plus countries. We also have a strong presence in US, Europe, and APAC. Good evening, Dr. Li. How are you today?
Very well. Thank you very much, Mark. It's a real pleasure to have this discussion. Hello, everyone.
Great. Great to have you. We're going to start with a couple of questions before your company presentation, and then we'll open it up to the audience for additional questions to close out the webinar. Thank you very much. So we'll start. Dr. Li, what inspired you to bring DNA encoded library DEL technology to China and establish HitGen? And how has your vision for the company evolved since it founded in 2012?
Okay, thank you very much, Mark, for that question. Obviously, it's a little while ago, and what actually inspired me at the time, it sounds a little bit vague now, but when I reflect on this, it's to do with my own experience before I founded HitGen in 2012. I worked in biotech and big pharma in the UK for over 22 years. And all that time was really about innovative discovery, creating new molecules to address different disease requirements, as well as discovery requirements. And I was all together before I founded HitGen in 2012. I was already outside China, studying and working for like nearly 30 years. Now, what happened during 2000 and 2010, there were quite a clear trend of emerging startups, biotechs, CROs in China, founded by many Chinese compatriots studying in the US and Europe and UK, returning back to China to start with this whole new wave of companies. And of course, I noticed that trend. And probably through various conferences, I got to know a little bit more about what's happening in China. And it was quite clear there was a transformation going on in China in terms of pharmaceutical industry. It used to be primarily dominated by generic companies, with obviously serving the medical needs of a large number of people in China. But there was also a growing trend of companies starting up, exploring new targets, new biology, and wanting to create new molecules and new medicine. Because of my own background in the early drug discovery, focusing on discovery technologies, creating new molecules for high throughput screening, I felt maybe as that trend continued, there would be a demand for new molecules. As it happened during that time, I would say, around 2005 and 2010, there was quite an immense effort going on, even in the big pharmas, about creating larger screening compound collections, as well as exploring new technologies such as DNA encoded libraries, as well as fragment-based drug discovery to meet those emerging new demands. And when I scanned the space in China, actually Asia, DNA encoded library wasn't actually noticed. While even in the US and in Europe and UK, it was still early days of the technology. So I felt that was an opportunity to bring this technology into China, and actually we were the first company in Asia to implement DNA encoded library technology and start to apply in different drug discovery contexts. Yeah.
Excellent. Thank you. Here's another one. HitGen's DEL platform has grown from screening millions to now over 1 trillion. What have been the key scientific and operational challenges in building and scaling such a vast and diverse chemical library?
Yeah, that's a really good question, Mark. I think at the time when we were looking to implement DNA encoded library, the publicized screening collection based on this technology is probably about 100 billion or so by 2 or 3 companies. I will not mention them, but so it was in comparison to the traditional high throughput screening compound collection, 100 billion molecules were already quite a large increase, or several orders of magnitude increase in the molecular entities for screening. So when we actually started, I thought that because we have to really know how the technology performs, how much it can scale, we didn't set a really large, very sort of high bar. We say if we can get it to hundreds of billions, it would already be a fantastic achievement. And the scientific sort of challenge at the time was around molecular diversity, because many people felt this scale of compound library design and synthesis would be limited by the number of reactions and type of reactions and the building blocks you can use, hence limiting your chemical diversity, which at the time was quite probably well-founded sort of skepticism. As you know, many novel molecules are created by a wide range of chemical reactions, as well as a large number of building blocks to choose from. But because of the presence of DNA, not all chemical reactions used to create new molecules are compatible with the DNA, the presence of DNA. So one of the scientific challenges is to expand the chemical reactions used to create new medicinal chemically friendly molecules, which are in the same time compatible. So that was probably one of the key scientific challenges we have to consider and build on, use it to create this much expanded DNA encoded library. Second one is more logistic, because at the basic level, what the DNA encoded library is utilizing is the systematic compilation of many different building blocks according to certain chemical reaction scheme. And that could involve thousands of building blocks being combined systematically. And if we do that purely by manual approach without a good process control, it could easily lead to a lot of mistakes. And that mistake can amplify as you go from hundreds of millions to hundreds of billions. And that's obviously really, really important technical process control we have to introduce. Another one, of course, is the use of this DNA ligation process to ensure the barcode are correctly put onto each reaction step, eventually onto each molecule. So these are the things we have to really overcome. We have to use a team, create a team to explore different DNA compatible reactions, as well as the technological process to ensure fidelity of the molecules created and the correspondence between the molecular structure and the DNA barcode. So these are quite a lot involved to get it to from few millions to few tens of billions to like now over 1 trillion molecules we have been using routinely. Yeah.
Incredible. Thank you. Here's another one. With HitGen's strong track record of global partnerships and more than 500 collaborations, what do you see as the most important factors for successful international alliances in drug discovery?
Yeah, so that's really, because that really underpins having a good customer base or collaboration partner base is really important. I also like to use this opportunity to thank all our partners and customers sort of in different regions all these years working with HitGen and giving us your trust in progressing your projects and contributing to your discovery effort. And I think there are several of the factors. I suppose really is to connect with our partners, customers early on, really understand their needs and how our sort of technology and resources and capabilities can contribute to their discovery effort. And secondly, really have effective communication during the research processes, identify, understanding, recognizing problems, and solving the problems, and being upfront about the advantages and disadvantages of this kind of technology platform. And thirdly, actually have very clear, structured, well understood commercial sort of model and framework, which protect the interest of our customers as an absolute priority. So, for example, because the nature of this technology, vast number of molecules, but when you do screen one target, doesn't matter when do you screen the same library, and if the protocol is the same, the molecules coming out are exactly the same. So target exclusivity as a principle in applying this DNA encoded library technology to different drug discovery program is absolutely very important. And we have to be strictly disciplined in following that so the exclusivity principle. And secondly, as I said, molecules coming out on the same target are the same. So we have to protect the IP design, so the expectation of our partners. So the molecules confirmed coming out of our screening have to be perpetually exclusively licensed partner. So these are the, I would say, very, very important commercial discipline to ensure this sort of collaboration are, first of all, productive, effective, and also protecting the IP position for the companies working with us. Yeah, these are really, I suppose, probably key principles we have been adhered to.
Excellent. Thank you. As the biopharmaceutical landscape shifts towards biologics and advanced therapies, how do you see the role of small molecules and HitGen's technology in driving innovation and meeting unmet medical needs?
Yeah, that's a really great question. I suppose for all of us working in the pharmaceutical industry, the priority, so the first thought should be unmet medical needs and patient needs. Doesn't matter what technology we adopt, and that has to be the number one concern. Then it's to say whether our particular angle of working in that industry, either it's small molecules, peptides, antibodies, or bispecific antibody, ADCs, radiotherapies, thinking there a whole range of siRNAs, a whole range of collections, probably is about so understanding the advantages and the limitations of our own individual technology, and finding the appropriate applications. That's one thing. Another one, of course, is keep innovation going on. In the first phase of encoded library, it was primarily focused on the traditional small molecules, the heterocycles, which can be used for oral administration. But actually, when we started to look deeply into this technology and its potential applications, peptides, cyclic peptides, macrocycles, all sorts of those things can also be sort of applied to. So that's why encoded library today has been adapted to address different emerging needs in addition to traditional small molecules, macrocycle, cyclic peptides for therapeutic discovery. Another application which has emerged, for example, for addressing the needs of siRNA, nucleic acid based therapeutics, RNAi. A key issue is around the delivery. And of course, there is a couple of predominant molecule-based delivery for siRNA applications, but there are many other emerging needs. For example, how to get siRNA to different tissues and cell types, in addition to the liver, sort of as a target organ. But others, lungs, sort of heart and brain, these all potentially have applications of siRNA-based drug molecules. In those areas, the DNA encoded library, peptides, cyclic peptides could also be used to discover delivery molecules for moving these molecules towards this target tissues and target organs. Yeah, so this is why we have to keep up innovation and really understanding the developing trends and adapt our sort of approach technologies to address those needs.
Thank you very much. We have one more question before your company presentation. Looking ahead, what are your strategic priorities for HitGen's growth over the next 3 to 5 years, particularly regarding in-house drug development, technology expansion, and global market positioning?
Yeah, so very, very, very good question. Clearly, this obviously is something we continue to think about, evaluate, and position ourselves. First one, of course, is to build on what we have already sort of established in the drug discovery space, our DNA encoded library, fragment-based drug discovery, and the emerging areas like targeted protein degradation and oligonucleotide-based therapeutic. These are the areas which we have already shown we have established effective technologies and already developing customer partner space. So our number one priority is continue to enhance and improve what we have in this sort of area, so we can deliver better and faster and more cost-effective to our sort of partner needs. And the second one, of course, is to explore the applications of, for example, encoded library technology on some of the emerging new targets, particularly in the areas where HitGen have felt there is a clear emerging need, for example, in oncology, autoimmune, immunology, inflammation. These sort of areas, there are a lot of opportunities in addition to support our customer needs, partners' needs, and with the principle I mentioned earlier, target exclusivity. We also can use the technology to develop some assets for out-licensing, collaboration. And in oncology, because we are largely a China-based company, we also feel there's a real demand and unmet needs in the cancer space. So we actually intend to develop selected assets in China and for patients in China. So that would be our second strategy. The third angle is obviously looking for sort of growth based on external strategic collaboration, for example, through effective mergers and acquisition to extend our capabilities, our business areas, and our reach in the market. And the final one, obviously, is to maintain and adhere to our sort of collaboration model, both internationally and domestically, to ensure our technology resources and capabilities can contribute wider effort in the innovative medicine discovery and development.
Great, excellent. Well, thank you very much for those intro questions and answers. We learned a lot. So please share your screen to start your company presentation. After that, we'll take questions from the audience. Thank you.
All right, thanks, Mark. I think now we'll show get our sort of slides. open the screen. Can you see it?
Yep.
Okay.
There you go. Thank you so much.
Thanks. Yeah, thanks very much, Mark, for this opportunity to really have this presentation and sharing overview of HitGen. So HitGen was, as Mark mentioned earlier, was founded in 2012, and our headquarters is in Chengdu, as you see from the PPT, that the brown reddish building in the International Biotown, 10th Biotown here in Chengdu. And we also have, in addition to this Chengdu base headquarters, we also have subsidiaries in Cambridge, UK, called the Venalis, as well as HitGen Pharmaceuticals in the UK and then US. We currently have about 500 people working in the company. As I mentioned, we come from what's called discovery technology platform-based company with a DNA encoded library as best known. And through Venalis, and they have been in this field for like nearly 30 years, developing and applying fragment-based drug discovery approach. More recent development in HitGen using our experience in DNA encoded library, particularly nucleic acid chemistry, is the oligonucleotide-based therapeutics, as well as adapting their technology to the protein degradation space. These are our primary 4 technologies. In addition to this, obviously, we have other medicinal chemistry, biology, in vitro, in vivo biology to really support all this effort. As I mentioned, we also, on a selective basis, pursue some in-house programs in oncology, in autoimmune and inflammation. We became a public company in 2020 on the Shanghai Stock Exchange. So company is organized into what we call the units. We have a discovery chemistry unit, which is our primary chemistry-based function with about 250 people. Anywhere from the designing of molecules, including libraries, to making, to medicinal chemistry, as well as analytical chemistry and scaling up. So all chemistry functions are in there. We also have what's called discovery biology unit. This is where all the dose screening or other in vitro, in vivo biology, pharmacology, and the early safety assessment are sort of housed here in Chengdu, over 90 people. And then we have our in-house project unit, we call the discovery project unit. This is more in-house program management unit, which nominates targets, progressing them to a stage which can be shared with our partners or enter into clinical trial. The Venalis is a fragment-based, it's very much self-contained from target protein science right up to sort of lead candidate generation. And they have been in fragment structure-based drug discovery space for nearly over 30 years and are very experienced group of people in that field. And this is our team. I'm not going to walk through every one of them, but do represent a wide-ranging background of people very experienced in the industry, in academia, in the relevant field. What we do is going really following the industry process from target to lead to preclinical candidate and to IND, and gradually build up our capabilities for each phase. And what we have been doing over the last 10 years, really to build this core 4 technology platform to support the transition from target to lead, advanced lead generation. We feel we have done quite a lot through the build up of DNA encoded library, FBDD, and TBD, and OBT. So these are the ones we feel quite well established and have a good partner base for its continued application and enhancement. What we also have built up capability is also leading out, going from the lead compound to preclinical candidates, as well as then the IND labeling studies. This is the broad guideline, which are obviously the industry standard process, guide our capability development as well as applications, and build up our expertise and resources. So I'm not sure the video will show on the screen, but this is a broad, sort of very simplified overview of the encoded library technology and the concept, which basically on the right-hand side is what we call the small molecule chemical compound through a linker, which is then link into a stretch of DNA. And of course, the different segments are colored differently of the DNA, which are used to represent what are the building blocks used in creating the molecule, in making the molecule, as well as the sequence of those building blocks are put together to form the final molecule. Making this one molecule is probably it's limited in its application, but when this basic concept is combined with making vast number of molecules through the split and pull combinatorial chemistry making process, it becomes a very sort of powerful in addressing a large number of molecules creation as well as screening. And because of the time limit, I will not go into details, but really this is a technology combining the power of combinatorial chemistry with the power of DNA encoding, information encoding. Not only can be used to create a vast number of molecules, as we mentioned earlier, we're already into over 1 trillion molecules of a very large number of building blocks and scaffolds, but also can facilitate effective sort of screening. What you can imagine is now, instead of sort of biologically testing individual molecules' activity towards a target, and the whole collection of molecules, including molecules, can be incubated with a targeted protein, and let those individual molecules compete and finding out the sort of high affinity ligands or a wide range of affinity ligand towards your target. And through the DNA sequencing, then we can identify which target molecules from the large screening collection are actually true binders to the target protein. So through the effort we have put in over the last 20, I must say it's quite a large effort. This is what we currently have, about over 1.2 trillion molecular species we have made in our labs, which cover quite a wide range of molecular types, but the majority is on the left-hand side, what we call the traditional novel heterocycle molecules for a variety of targets. But what the technology can clearly be adapted to is also making macrocycles or cyclic peptide molecules. And this is also quite emerging as a major area of interest from our sort of discussions with our customers and partners. Another type of molecule which could be difficult to imagine this can be used to is a covalent small molecule discovery, or covalent to some other types of molecule can equally be applied. And PROTAC or bifunctional, multifunctional molecules can also be can be created using this sort of approach. Through our sort of acquisition of Vanalis, we also realized, because they specialize in fragment-based drug discovery, traditional fragment drug discovery collection is very small, usually in a few thousand of fragments. Clearly, we believe that's a limitation in the use of FBDD. So we adapted sort of DEL technology to create what we call the pack fragment DEL, using DNA encoded library approach to create tens of thousands, even hundreds of thousands of fragment molecules. So these are our primary sort of screening collections we use at HitGen for our customer projects. In order to expand the use and support a variety of research, some sort of biotechs and academic groups, we also launched OpenDAO a few years ago. This is a smaller collection with about 3 billion molecules in there, which can be purchased and used in your own laboratories to screen novel targets of interest. So this is a range of DAO libraries we put on there to support the activities. This is a summary of how these sort of libraries have been used and what's its performance based on last 10, 12 years screening against the different target types. These are, as you can tell, a variety of target types. There are some like over 50, some different target classes and different distribution. As you can see, the largest collection of targets and types we worked on is a protein-protein interaction, which are long traditionally very difficult to find the small molecules disrupted the interaction between the PPI, so-called the protein-protein interaction pairs. And usually they are called the wrong drug, undruggable or difficult to drug target class. As you can tell, there's a large class there we're working on. There are also more traditional ones like kinases, the orange bar, and many, many other emerging novel target class or traditional target class as well. And on the top right-hand corner of the bar is the performance of the screening we have sort of generated. The orange bar is the number of targets we screen in that year, and the green is the percentage of success coming out of the screening. We call them based on, for example, confirmed hits with activities less than 10 micromolar. And as you can tell, also the blue bar represents the confirmed active, so the targets with the confirmed hits, which are permanently licensed to the partners. As you can tell, it's a very steady growing trend over the years. And the bottom right-hand corner really reflects the activity range of those confirmed hits. The range from 10 micromolar is a cutoff. Majority are below 1 micromolar. Many are those in the tens or hundreds of nanomolar. So this is a quite an effective tool in generating hits for a variety of targets. So based on our sort of continued effort in the DNA encoded library space, and it's become clearly recognized in the industry, particularly by those big pharmas. So in 2024, 6 of the top big pharmas, Pfizer, Johnson, AstraZeneca, Roche, BMS, and Merck, formed what we call the DAO consortium with HitGen, and to continue to explore this technology to meet their future drug discovery needs, as well as to combine in a non-competitive way, a pre-competitive way, of key resources and expertise and knowledge and know-how from these 6 pharma companies and HitGen together to create a high quality and really continue to push the boundary of this technology for the discovery needs of the future. Another important sort of initiative we are very honored to be part of is this Structural Genomics Consortium, which aim to so generate something like about 2,000 novel proteins between 2025 and 2035. Another important goal of that initiative for the protein that generate during that initiative, that 10-year period, is to find ligands for the target generated. And we are very honored to be one of the partners contributing the chemical libraries to screen against those targets they generate under the Structural Genomics Consortium. And final one, which we felt also as part of that initiative, is very important. And many people know AI drug design or drug AIDD is a very important emerging, potentially very high impact sort of field, is to using AI to support the drug design. And one of the key factors for this approach to become more impactful is data. So one of the key output from the SGC is those sort of target and compound screening data will be put into the public domain to support the testing and development of the new AIDD algorithms and approaches. So we feel very sort of honored and sort of privileged to be part of that consortium as well. So finally, as you can tell, we feel the Dell technology can be applied in many sort of different areas as well, different contexts. Last year in 2024, we also joined a program launched by Mission Bio Capital in the US. This is about really providing early funding and early sort of discovery support for those sort of innovative startup companies from start. We will also be part of the sponsors to get this some of the really creative startup companies to be able to access this technology as well. Another movie, yeah. Okay, so that's just a quick sort of description of what the other aspects of HitGen's platform is like. So we have this, as I mentioned, through Velanis, we have a FBDD, SBDD platform. This is aimed at really using a very different approach from traditional HTS or Dell to base on fragments, the smaller pieces of molecule building blocks to as a starting point to bind it to particular protein, then combined with a rapid structural biology approach to provide information on how the molecule binds to the target to support the medicinal chemistry design and optimization of the molecule to become a candidate, a drug from fragments. And protein degradation has become a really major focus in the drug discovery space over the last few years, and some of them have got into clinic. Many more programs are in preclinical stage. And when we looked into this field, it was quite clear the opportunities will lie in many different directions. Of course, exploring well-known E3 ligases already and novel ligand discovery there, but also exploring new E3 ligases, which are still unknown. And what is in the public domain currently with the non-degradation capacity of E3 ligases is probably less than a dozen in the public domain. But through the genomic and other analysis in human body, there are like over 600 E3 ligases. Many of them are of unknown degradation in terms of targets and the tissue distribution and what's the balance between degradation and other effects in the biological system. So HitGen right from beginning felt what in order to support the discovery in that space, we must explore a novel E3 ligases. So we expressed about 50 different E3 ligases, which then we undertake Dell screening to identify which ones could become new E3 ligase leading to degradation. So this is a whole lot of resources that have been built up, which some of them have become published or coming into notice. One of them is TRIM21, and we have been able to find a novel ligands and molecular glue to show its degradation. And clearly we need to explore a lot more of that space. Because our effort in the DNA encoded library, a lot of activity was built up to ensure the fidelity of this DNA barcode. So hence, our nucleic acid chemistry capability were sort of tuned over the years to be really effective and high quality. As many people know, recent years, although this field has been around for many, many years, but with the sort of clinical success of PCSK9 sort of siRNA therapies due to their longevity of duration of action, there is a rapidly growing interest to discover more siRNAs or other forms of RNAis. So we felt our expertise and skills in nucleic acids could be built up and transformed to support that area of research. So we have built this sort of line of activities from design, synthesis, evaluation, and eventual GMP manufacturing of siRNA or nucleic acid-based drugs to support the industry. So as I mentioned, what we have been doing is really building capability and resources to move from targets to leads. But what it needs to be continued to improve on is how, once we find the early hits or leads, how can we change or optimize them into preclinical candidates? Traditionally, this is called the lead optimization, based on design, make, test, analysis cycle iteratively over a period of time to achieve the target product profile of the program. Many of the established approaches really is driven by experience as well as resources commitment to make molecules and testing in various variety of biological models and PK evaluation and early safety assessment. As AI become more and more applied into drug discovery space, we felt this could be an important development in improving the lead optimization process based on these design, make, test, analysis cycles. And one of the key elements is, for the use of AI is whether there is sufficient high quality data to train the models. So the first approach we consider where we come in, because after dose screening, we do produce a lot of molecular evaluation outcome on whether they bind to the target. So that data could be very useful, the dose screening output, to generate predictive models on new molecules which whether binds or not. But making larger number of them requires sort of maybe automation. So we build up a automated chemistry lab which is capable of making over 10,000 molecules per month. And once those molecules are made, can we produce enough data rapidly to feed into next cycle of analysis and model building? So this is what we have built up already, and what we call the Halo platform. So high throughput AI-driven lead optimization platform as our next extension of technology platform in the value chain of drug discovery. And hopefully in the next a few years, we'll apply this to many projects and generate sufficient data to show how much improvement this process can lead to our lead optimization. So as I said, in addition to what we built in technology platform and use them to support our partners program discovery effort, we also apply the technology capabilities in a limited way to oncology and autoimmune field. And this is our portfolio. Some of them already in clinical trial, other in preclinical stage. Our most advanced sort of program is HG146, which is HDAC1, class 1, class 2B selective molecules, currently in clinical trial for adenoid cystic carcinoma, which has been shown to discovered in phase 1 trial have a clear, so effective response. Hopefully we'll be able to complete the phase 2A program this year and report how well the molecule perform in that particular indication. So just a summary of our business model. We classify into two arms. One arm is on the left-hand side, which really are traditional or bundled sort of services, chemistry, biology services, as well as there's a full platform-based we call the bundled technology services. On the right-hand side is more about the program-based collaboration, either based on some programs that we have already moved at a different stage of discovery and development, or actually from beginning to do what's called integrated drug discovery, or form new joint ventures to use our technology to address needs in particular therapy area or pathways or target classes with our partners. Okay, this is just a glance of our publicized already in the public domain partners. We have over 500 sort of customers and partners working with us globally, and we hope to grow that in the next few years onto like 2,000. And it's really a pleasure, and it's also giving me a great sort of honor and privilege to thank all the partners over the last 10 years to work with HitGen. I hope our collaboration continue. Okay, this is where we are in the Biotown in Chengdu on the left-hand side. On the right-hand side is our new research headquarters being built, also in Biotown here in Chengdu. And you're all welcome to visit us, and we are in China next time. Thank you very much.
Great. Thank you, Dr. Li. Thank you for the accompanying presentation. So let's gather some questions from the audience. I believe, so you described HitGen's core technology platforms, the DNL encoded library, the fragment-based drug discovery, synthetic therapeutic, oligonucleotide, and the targeted protein degradation. How do they differentiate HitGen in the global drug discovery landscape?
Yeah, so it sometimes, as you know, it's very difficult to describe this so differentiation in sort of simple one-liners. But in terms of encoded library side, I would say based on the published data, HitGen's encoded library probably represents the largest small molecule library for screening, and based on vast number of novel scaffold, wide range of chemical reactions, as well as large collection of building blocks. So, and another one is we have publicized lots of sort of encoded library collaborations, as well as being able to demonstrate the already highly stable success rate over a wide range of target classes. I think that would probably be the key differentiator we have for encoded library. For FBDD, SBDD, and of course, this is a more established area in drug discovery space. And for the company of our type, this kind of size, FBDD primarily through our subsidiary Venalis in Cambridge, UK, are probably one of the most sort of experienced team in the industry, which can really be sort of form a partner with either big pharmas, medium pharma, or biotechs for the drug discovery. As a technology which is well tested and validated in its ability to generate high quality candidate molecules, because they start with a small fragment. So molecules can be really carefully engineered to meet all the drug-like properties in many different contexts. And they have published many sort of papers, as well as delivered clinical candidates with the partners over the years. And oligonucleotide therapeutics, this is again rapidly developing. What I would say, we have been one of the fastest growing company in this field for providing the fully integrated capabilities from design, high quality synthesis, as well as the biological evaluation, and eventually to the GMP manufacturing. So there's a full line has gone through. I guess another addition in this field, differentiation we have, is our encoded library resources are now being actively applied to discover new delivery molecules for siRNAs to not only liver, but into other organs as well. Degradation, I would say one key differentiation we have is probably exploring the wide ranging of E3 ligases, which are unknown in the public domain, through our, for example, recent publication on TRIM21, which not only showed has a degradation through molecular glue, but also through PROTAC. And so equally there, DEL has played a very key role in discovering new binders for new POIs, as well as even discovering new ligand for established E3 ligases, for example, the CRBNs and some other announced E3 ligase identified by our partners. So we do have significant differentiation in each one of those fields. My guess, if you want to really bring down to one key thing, is really the molecule. Because for us, creating new molecules in each of this platform has been the heart of our effort. Also, I believe that could form differentiation for us as well.
Great. Here's another one from the audience. How has HitGen's global presence with headquarters in Chengdu, subsidiaries in UK and USA contributed to its innovation, operational capabilities, and international collaborations?
Yeah, I think maybe starting with the last one, because many are maybe through that slide I shared with you early on, many of our partners are actually international biotechs, pharmaceutical companies, research institutions, as well as foundations. So our presence in UK through Valaris, which is obviously linking to Europe and the UK, and also our colleagues in US have been playing really effective role in driving, supporting, identifying those collaboration opportunities and ensure effective communication with the partners. Other aspects are innovation. I think innovation, there are many opportunities for innovation, and in our industry, innovation probably I usually think is best innovation are the ones that effectively address our partner needs and new scientific opportunities. Another one is, of course, internal collaboration. Using fragment as an example, Valaris has been well known and very experienced in fragment-based drug discovery through the really effective biophysical characterization of those small fragments, and then using effectively the structural biology to drive the medicinal chemistry design. But the fragments they used to use for their screening was rather limited. It's like a few thousand fragments. But combining with HitGen's DEL early on, the two groups here in Chengdu and in Valaris, Cambridge, UK, discussed and used DEL technology to expand that fragment collection from a few thousand, a few tens of thousands, maybe hundreds of thousands in the future. And that clearly contribute to the massive, I would say, rapid expansion of fragment diversity for use. Another important advancement there through that effective collaboration and innovation is fragments tend to be very weak in binding into tens or even hundreds of micromolar. And naturally that leads to the large protein consumption in order to test that binding. But with the encoded fragments, because the DNA's PCR, you can amplify with very small quantities of the molecules. That also directly contribute the really much huge reduction in the protein consumption in using this kind of DNA encoded fragment library for screening. So I would say innovation, that's already one of the good examples. There are some others. As time goes by, we're going to be able to share a little bit more. Operational capability is clearly each group has different strength, but also where we can complement each other. We do complement, not only driving for quality output, but also for cost-effective operations to really translate that effect to our partners as well.
Great, thank you. So I think you sort of touched upon this, but maybe they want to dig in a little bit more in terms of what are the main therapeutic areas and disease indications HitGen is focusing on within its internal R&D pipeline, and what are some notable assets currently in clinical development?
Yeah. Okay, thanks. I think, as I mentioned earlier, we are very selective because we are still a fairly small company. We can't sort of spread ourselves too thinly. So we are mainly selecting opportunities in oncology, and that we aim to sort of develop oncology drugs in China. But the external China rights, we aim to out-license to international partners. Another area which clearly has large unmet needs, as well as rapidly developing new mechanisms and targets opportunities, is inflammation or autoimmune. And these are the primarily two therapeutic areas we focus our effort on. For the oncology space, as we mentioned earlier, our advanced asset is a selective HDAC inhibitor in phase 2A studies. And we did have early clinical phase 1 compound in STING, so innate immune or against cancer. Preclinical data all showed very promising, but there are still challenges in developing that asset. One of the assets we already out-licensed early on now in clinical trial in China is our sort of NTRK ROS1 inhibitor that was licensed to China-based BioInsan. And that asset we actually have already valid FDA IND approval for international trial. Apart from this publicized or target I disclosed, we still have a few other programs in the preclinical or early discovery optimization phase, again, in this space. And we don't have a large portfolio. We have at any time active programs, less than maybe a dozen. So we are highly focused in those areas and hope to develop opportunities for collaborations in the next few years.
I guess on that, how does HitGen business model balance the research service offerings, out-licensing, co-development partnerships with global biopharmaceutical companies?
Yeah, that's very, very good. So I think the balancing is definitely, as I mentioned earlier when we have very strong discipline, that is the target exclusivity. And for all our targets must be free from the exclusivity, so that they are not occupied by our customers and partners. And the ones we are working on, we disclose to our active partners so they know what we are working on. So to ensure there is no contamination or conflict of interest. In terms of resource commitment, we in general use like 15-20% of our revenue to fund internal research. And that covers two areas. One is the continued technology platform innovation, improvement. Another one is those in-house programs. So that's all covered by our internal R&D investment.
Great. What has been the most significant recent milestones or achievements for HitGen in terms of technology advancements, global partnerships, or financial performance?
Yeah, I think I would say what we have done as a public company since 2020 is a steady, continued growth in revenue and profit. So that's a financial performance. Technology advancement, over the last few years, I think as I mentioned already, bringing encoded library approach into fragment space, the discovery of truly novel E3 ligase binders, and the validation of them in degradation in certain tissues and targets. And global partnerships, as I mentioned, I think probably the highlight are those three, which I mentioned. One is with a 6 big pharma, forming the DEL consortium launched in 2024, as well as entering, being selected as a partner for the SGC targeted 2035 effort, as well as the collaboration with the capital investment capital group, Mission Bio Capital, in sort of identifying and supporting early innovative discovery startups.
Great. I just have two more. I don't think it's part of the trend here in terms of the AI question, but how is HitGen leveraging artificial intelligence and machine learning to enhance DEL screening, drug discovery efficiency, and data analysis?
Yeah, this is clearly a very important area and potentially have very high impact as we go into the future. But as people involved in this whole field knows that really some of the key elements supporting its application are: one is the computing power, and secondly, computing algorithm, and thirdly, the data feeding into the AI system. So we have really been using AI machine learning approach in a different way for several years now. As I mentioned, we involve a lot of, in our process of making encoded library, we have to involve the compilation, synthesis, and analytical assessment of many, many molecules per day or per week. And being used to using human, so eyeballing of this analytical data, it takes quite a long time and sometimes can make mistakes. So several years ago, we trained a machine learning algorithm to do that job. That really has been used for several years without now even being noticed. So it has embedded into that particular part of the process. Another one is use our accumulated DEL screening data to train models for predicting accessing other chemical space. And that we did some early collaboration. We also continue that effort and already are using some of that in our ongoing services with our sort of customers. I suppose that area will continue to expand, continue to be explored. As I already alluded to, in our sort of next wave of platform development is this Halo platform, which will be based on starting with a DEL output, screening output, and generate a predictive model and also generate new molecules for rapid synthesis by automation labs or even manual synthesis, and then rapid high intensity data generation. And that feed into analysis and model building. Hopefully that would drive improvement in lead optimization. So that, I think this is a continued area of investment, exploration, and application. Through that consistent, continuous application, feedback, and improvement, we hope we can really develop a more differentiated offering in this field.
Well, congratulations, Dr. Jin Li, on your discoveries and all accomplishments to date. Thank you for sharing all your knowledge to get a better understanding of HitGen and your firm's scientific advancements behind your leadership. Any last words for the audience before we conclude?
Okay, thank you, Mark. Thanks very much for this opportunity to really share our journey, our thinking, and where we've got to for HitGen. And really, discovery research is such an important effort as it is the foundation for address series unmet medical needs. And the key thing there, of course, is continued innovation. And there is so much needs, and also the so uncertain process. The use of new science and new technology are fundamental to the success of that effort. HitGen will continue to focus on the areas we have identified and also continue to improve our sort of science and technology operating processes and working closely, openly and transparently with our partners to deliver better outcomes and more effectively in the future. Many thanks for our customers and partners' trust and collaboration over the last 12 years, and continue to hope we can continue to build on this collaboration and address serious unmet medical needs and help contribute to discovery and development of new medicines in the future. Thank you very much.
Thank you. We are excited and eager to see HitGen's future milestone achievements. Thank you everyone for your interest today in HitGen and in MSQ's CEO Founder webinar series. Have a great day and good night.